Immunoprecipitation assays use antibodies and solid substrates to specifically enrich and purify peptides, proteins, or protein complexes from biological samples, usually cell line or tissue lysates. Following immunoprecipitation, Western blot, mass spectrometry, or other techniques may be employed to analyze the peptides, protein, or proteins of interest. Immunoprecipitation experiments can be used to detect and quantify the presence of lower abundance proteins, assess changes in protein post-translational modifications such as phosphorylation, or elucidate protein-protein interactions, among other experimental aims. This video demonstrates the immunoprecipitation protocol for native protein for Western blot analysis. The immunoprecipitation protocol is organized into sections, and this video is broken into corresponding chapters, which can be accessed using the timeline at the bottom of this video. We recommend that first-time users make sure to watch the Protocol Considerations chapter, which covers tips for experimental design, controls, and advice for choosing reagents. You can also follow along with the written version of the protocol, available on cellsignal.com and linked below this video. Related methods not depicted in this video include co-immunoprecipitation, peptide immunoprecipitation, or motif-based enrichment. These can be used to study protein-protein interactions, protein complexes, and intracellular signaling activities. Immunoenrichment may also be used to prepare biological samples for mass spectrometry-based proteomics analysis. Rather than reading out a single protein target, these studies can monitor protein activities, expression, or dynamics across a broad portion of the proteome. These alternate methods are described in more detail on the Immunoprecipitation Experimental Design Tips webpage. This resource is located in the CST Immunoprecipitation Resource Center, where you can also find information on antibodies, controls, and companion products, a troubleshooting guide, and links to technical support. Visit anytime using the links below this video. Before designing and setting up your experiments, let's review guidelines for selecting antibodies, beads, and controls for immunoprecipitation.
Experimental design and reagent selection for immunoprecipitation assays are interdependent and are based on the desired protein targets and readouts. When identifying protein targets and selecting antibodies, check online resources such as biogps.org, fossilsite.org, or gene and protein expression databases such as the Human Protein Atlas. These can provide information about protein expression levels in your cell or tissue model and help you understand what to expect from your results. Application-specific validation data for CST antibodies can be viewed by searching the product number on cellsignal.com. Both protein expression levels and protein modifications, such as phosphorylation, ubiquitination, or other post-translational modifications, may be affected by experimental conditions and the overall health of the cell or tissue sample. If the cells or tissues are being treated with activators or inhibitors as part of the experimental design, carefully consider the treatment time point and concentration, as small changes could potentially have an impact on the readout.
Single protein immunoprecipitation involves enrichment of the target protein followed by readout of the same protein.
Select an antibody to the desired target protein that has been validated for use in native immunoprecipitation. Most of these antibodies enrich total protein or peptides, including unmodified and post-translationally modified isoforms. Alternatively, IP-validated antibodies that only bind protein targets bearing phosphorylation or post-translational modifications may be used to specifically immunoprecipitate proteins or peptides bearing these modifications. These are particularly useful for assessment of intracellular signaling activities. Most commonly, unconjugated antibodies are used in conjunction with protein A or protein G beads. Alternative approaches use biotinylated antibodies with streptavidin beads or antibodies directly conjugated to beads. All of these variations are demonstrated in section C of this video. The same antibody used for IP may also be used as the primary antibody for Western blot detection, as long as it is also validated in Western blot. If not, a different Western blot validated antibody to the target should be selected.
Now, let's look at the options for beads, which serve as a solid substrate and allow separation of target proteins from the bulk sample. Select protein A conjugated beads for unconjugated rabbit IgG antibodies, or protein G conjugated beads for unconjugated mouse IgG antibodies. Alternatively, immobilized antibodies that have been conjugated to beads or biotinylated antibodies and streptavidin beads may be used for immunoprecipitation. Refer to the IP experimental design page for considerations. IPs can be performed using either agarose or magnetic beads. Agarose beads are used to separate proteins from the bulk sample by centrifugation, while magnetic beads are separated on specialized magnetic racks. Cephalose is a trade name for agarose beads, and we will refer to agarose and cephalose collectively as agarose in the rest of this video.
Both agarose and magnetic beads are widely used for protein immunoprecipitation in labs around the world, and by scientists at CST during antibody validation testing for immunoprecipitation. Both separation methods exhibit similar performance. Your choice may come down to what is already stocked in your lab or personal preference. Helpful tips for the handling and washing of beads are included in this video to help you minimize loss of the target protein. The inclusion of controls can be used to optimize and troubleshoot IP experiments and to appropriately interpret your IP results. These controls are run as adjacent lanes on the protein gel for Western blot analysis. CST recommends including 3 controls in every IP experiment: input lysate, isotype control, and a bead-only control. First, an input lysate control is used to confirm that the protein of interest is expressed at sufficient levels and that the Western blot detection is working correctly. Prior to Protocol Section C, immunoprecipitation, remove an aliquot of the prepared lysate and reserve for loading the input control lane for your protein gel. An isotype control is a necessary negative control. It is used to report if non-specific binding of the target of interest to the antibody is occurring. Choose an isotype control from the same host species and immunoglobulin class and subclass as the antibody used for the immunoprecipitation reaction, and match its concentration to the IP antibody. A bead-only control is recommended to detect if non-specific binding or absorption of the target protein is occurring. After the beads are pelleted the first time by centrifugation or magnetic separation, you may save the supernatant for optional analysis. This contains the unbound fraction of the target protein. The unbound protein fraction can be compared by Western blot to the enriched fraction bound to beads to determine the efficiency of the IP reaction, and as a troubleshooting step to indicate if the target protein did not bind the antibody bead complex. The preparation and handling of lysates, demonstrated in Protocol Section B, can also impact the success of your IP experiment. For optimal IP results, use freshly prepared lysates. Lysates may be stored at -20 for a month or more depending on the protein, or at -80 for up to a year. Note that some proteins are more sensitive to degradation caused by freeze-thaw cycles, while others are more stable. To reduce target degradation, include protease inhibitors in the lysis buffer, and add phosphatase inhibitors if needed for your target. Lysis buffer containing protease inhibitor cocktail may be prepared ahead of time. However, the 1 millimolar PMSF should be added immediately prior to use. For efficient protein extraction and consistent lysate quality across experiments, brief sonication of lysates followed by centrifugation is recommended. A probe sonicator rather than a water bath sonicator is preferred.
Before beginning the immunoprecipitation experiment, prepare the following solutions using reverse osmosis deionized water or equivalent grade water. 1x PBS prepared from a 20x stock and placed on ice. For immunoprecipitation of native protein from cell samples, we recommend 1x cell lysis buffer. Immediately before starting your experiment, dilute a 200 millimolar stock of PMSF, 1 to 200 in cell lysis buffer, for a final concentration of 1 millimolar PMSF. If desired, additional protease inhibitors or phosphatase inhibitors may be added at this time. For information on lysis buffers for tissue samples and other immunoprecipitation protocols, please refer to the Immunoprecipitation Experimental Design Tips webpage linked below this video. You will also need to prepare 3x SDS sample buffer by adding 1/10 volume of 30x DTT to 1 volume of 3x SDS loading buffer. Choose either agarose or magnetic beads, unconjugated or streptavidin conjugated, as described in the Protocol Considerations section. When setting up your immunoprecipitation reactions, the bead stock should be kept on ice. For all immunoprecipitation protocols, you will also need the following equipment: A probe sonicator. A mechanical rotator located in a cold room or refrigerator. A refrigerated microcentrifuge capable of 14,000 Gs. A magnetic separation rack if using magnetic beads. And a heat block if using Western blot analysis. Let's begin the IP protocol for native protein, starting with preparation of cell lysates. We will demonstrate with the adherent cell line HEC293.
For native IP using cell samples, we recommend lysing the cells with cell lysis buffer. For IP-related protocols, review the Immunoprecipitation Experimental Design Tips webpage for lysis buffer recommendations.
If performing cell treatments with activator or inhibitor, first aspirate the media from the cells. Treat cells with media containing the regulator, activator, or inhibitor for the desired time in the incubator. Remove media containing the treatment and rinse cells once with ice-cold 1x PBS. Aspirate the PBS and add 0.5 milliliter ice-cold 1x cell lysis buffer per 10 centimeter plate, and incubate on ice for 5 minutes. Harvest the lysed cells by scraping the plate and use a pipette to transfer to a microcentrifuge tube. Keep the tube on ice. Sonicate using a probe sonicator, 3 pulses for 5 seconds each. Adjust the power on your sonicator to avoid foaming the lysate.
Sonication of lysates helps to share DNA, reduces the viscosity of lysate samples, and helps ensure complete and consistent lysis from experiment to experiment.
Clear the sonicated lysates by centrifugation for 10 minutes at 14,000 Gs at 4 degrees in the microcentrifuge to pellet cellular debris. Transfer the supernatant to a new tube. This is the lysis sample. Reserve a portion of the lysate in a separate tube for use as the input control. If needed, store the lysate in a -20 or -80 freezer. Otherwise, proceed to the next section. Before starting the immunoprecipitation reaction, let's review some tips for handling bead stocks and suspensions.
Before transferring beads from the stock solution, invert or flick the bottle or tube several times until the beads are completely resuspended. Bead stocks may be vortexed, but once proteins are bound to the bead, avoid vortexing since this may cause the target protein to fall off. Use scissors to cut a pipette tip to make the opening slightly larger. Then transfer the desired volume of bead slurry. This helps ensure consistent quantities of beads in each experiment. During wash steps, take care to avoid aspirating the beads after they are pelleted. Loss of beads will reduce the quantity of immunoprecipitated target protein. Some users find it helpful to remove most of the supernatant using a P200 pipette, leaving a small volume of liquid, and then switch to a smaller volume pipette tip or a gel loading tip to carefully remove the last few microliters of liquid without disturbing the bead pellet. To minimize loss of magnetic beads during wash steps, keep the tube in the magnetic rack when pipetting off supernatant. To mix or resuspend IP reactions, gently flick or invert the tube, and do not mix by pipetting, which can lead to loss of beads.
Prior to setting up an immunoprecipitation reaction, an optional pre-clearing step may be performed.
Lysate pre-clearing is an optional step for protein A or protein G agarose beads, or for magnetic bead antibody conjugates. But pre-clearing is recommended if using protein A or protein G magnetic beads. Lysate pre-clearing can also be used to reduce the appearance of nonspecific bands if they are observed in Western blot analysis. The lysate is first incubated with beads in the absence of antibody, and the beads are discarded along with proteins that are nonspecifically absorbed. We will first demonstrate lysate clearing steps for the main native IP protocol using agarose beads, then demonstrate lysate clearing steps for the magnetic separation protocol.
Before pre-clearing agarose beads, measure the protein concentration of the lysate and dilute with cell lysis buffer as needed to adjust the final protein concentration to 1 mg per ml. Resuspend the bead stock by inverting or flicking the tube. For each 200 microliters of lysate, add 20 microliters of protein A or protein G agarose 50% bead slurry, or 10 microliters of streptavidin bead conjugate if using a biotinylated antibody for the immunoprecipitation reaction. Scale the volumes based on the number of reactions being performed. Incubate on a rotator for 30 to 60 minutes at 4 degrees. Centrifuge at 3,500 Gs for 10 minutes at 4 degrees. Carefully remove the supernatant by pipetting and transfer to a new tube, and proceed to immunoprecipitation. Before pre-clearing magnetic protein A or protein G beads, measure and adjust the lysate concentration.
The optimal lysate concentration will depend on the expression level of the protein of interest. A starting concentration between 250 micrograms per mil to 1 milligram per mil is recommended when using magnetic beads. Before pre-clearing and immunoprecipitation, magnetic beads should be pre-washed with cell lysis buffer. To save time, you can calculate the total quantity of magnetic beads needed for pre-clearing, plus the total number of IP reactions, and follow steps 1 and 2 of this section.
Briefly vortex the stock tube to resuspend magnetic beads. Transfer 20 microliters bead slurry reaction to a new tube. Place the tube on a magnetic rack for 10 to 15 seconds or until solution turns clear. Carefully remove liquid by pipetting. Add 500 microliters 1x cell lysis buffer and briefly vortex to wash the beads. Place the tube on a magnetic rack for 10 to 15 seconds or until solution turns clear. Carefully remove liquid by pipetting and repeat the wash step once for a total of 2 washes. Carefully resuspend the washed beads by pipetting up and down before using in pre-clearing or immunoprecipitation steps. Add 20 microliters of pre-washed beads per 200 microliters of cell lysate. Incubate on a rotator for 20 minutes at room temperature. Place beads on the magnetic rack until the solution becomes clear. Transfer the pre-cleared lysate to a new tube by pipetting and discard the magnetic bead pellet. Proceed to the immunoprecipitation coming up next. During the immunoprecipitation reaction, antibodies bind to their target antigens, forming immunocomplexes that are in turn bound to the bead substrates to allow enrichment. Slightly different protocol steps are needed depending on the type of antibody and bead used. We will demonstrate several different options in this section of the video. If using either unconjugated or biotinylated antibodies, first check the product datasheet or product webpage for the recommended dilution.
A common misconception is that a particular microgram quantity of antibody is required for all immunoprecipitations. For each individual antibody, CST provides the optimal dilution based on validation data generated using that antibody.
Add the recommended dilution to 200 microliters of lysate and incubate with gentle rocking overnight at 4 degrees to form immunocomplexes. If using unconjugated antibodies and agarose protein A or protein G beads, resuspend the bead stock by vortexing and add 20 microliters of 50% bead slurry to the lysate antibody mixture. Incubate on a rotator or with gentle rocking for 1 to 3 hours at 4 degrees. If using biotinylated antibodies, resuspend the stock of immobilized streptavidin cephorose bead conjugate by gently mixing. Then add 10 microliters to the lysate antibody mixture. Incubate with gentle rocking 2 hours at 4 degrees. If using unconjugated antibody with magnetic protein A or protein G beads, pre-wash the beads with 1x cell lysis buffer, if you have not done so previously. Refer to steps 1 and 2 of the magnetic bead pre-clearing section earlier in this video for instructions. Add the lysate antibody mixture to the pre-washed magnetic bead pellet and resuspend by gently pipetting up and down, and incubate with rotation for 20 minutes at room temperature. If using either immobilized antibody cephorose bead conjugates or immobilized antibody magnetic bead conjugates, first measure the protein concentration of the lysate and dilute with cell lysis buffer as needed to adjust the final protein concentration to 1 milligram per mil. Gently mix the stock of immobilized beads by inverting or flicking the tube and add 10 microliters of beads to 200 microliters of lysate. Incubate with gentle rocking overnight at 4 degrees.
After antibody-antigen immunocomplexes have formed and bound to beads, thorough washing of the beads helps to reduce background signal caused by nonspecific binding. To minimize loss of beads and immune complexes, we recommend using a pipette to remove supernatant, not vacuum aspiration. Keep the tubes on ice in between wash steps to minimize degradation. After beads are pelleted the first time by centrifugation or magnetic separation, reserve the supernatant containing the unbound fraction and store for analysis if desired.
For IP with agarose beads, microcentrifuge the reaction for 30 seconds at 3,500 Gs for 30 seconds at 4 degrees. Using a 200 microliter or finer pipette tip, carefully remove supernatant and transfer to a new tube. This is the unbound fraction. Store it at 4 degrees or on ice if analysis of the unbound fraction is desired. Wash the pellet by adding 500 microliters of 1X cell lysis buffer and resuspend by gently pipetting up and down. Centrifuge a second time, then remove and discard the supernatant by pipetting. Again, resuspend in 500 microliters of 1X cell lysis buffer and repeat the wash steps 3 additional times for a total of 5 washes. After the spin, refer to the sample analysis section of this video or to the experimental design tips page for the intended buffer used for the final resuspension. For IP with magnetic beads, place the tubes containing the IP reaction on a magnetic rack. Wait 1 to 2 minutes until the solution turns clear. Using a 200 microliter or finer pipette tip, carefully remove the supernatant and transfer to a new tube. This is the unbound fraction. Store it at 4 degrees or on ice if analysis of the unbound fraction is desired. Wash the pellet by adding 500 microliters of 1X cell lysis buffer and resuspend by gently pipetting up and down. Place the tube on the magnetic rack a second time. And after beads are separated, remove and discard the supernatant by pipetting. Again, resuspend in 500 microliters of 1X cell lysis buffer and repeat the wash steps 3 additional times for a total of 5 washes. After the final separation, refer to the next section or to the experimental design tips page for the intended buffer used for the final resuspension. This section of the video covers preparation for analysis by Western blot. If your IP reaction will be analyzed by mass spectrometry, refer to the Immunoprecipitation Experimental Design Tips page for instructions on preparing samples for liquid chromatography, mass spectrometry, or MALDI-MS. For Western blot analysis, after removing the supernatant from the final bead pellet from section C, resuspend the bead pellet in 20 to 40 microliters of 3X SDS sample buffer. Vortex to mix, then centrifuge the tubes for 30 seconds at 14,000 Gs. Transfer to a SafeLock tube, a screw cap tube, or use a cap lid lock. And place tubes in a heat block and heat at 95 to 100 degrees for 5 minutes. For agarose or Sephiroth beads, centrifuge the tubes for 1 minute at 14,000 Gs. For magnetic beads, place on a magnetic separation rack for 1 to 2 minutes until the brown bead pellet is separated from the sample buffer. After separation, transfer the supernatant, which should now contain the enriched, denatured target protein, to a new tube. Load 15 to 30 microliters of the IP-enriched sample onto an SDS page gel. Also, load lanes for molecular weight marker, input sample, and the unbound fraction sample saved from Section C, if desired. Please refer to the Western blot protocol and companion video for a full guide to Western blot analysis.
This concludes the protocol. Should you have any questions about the immunoprecipitation protocol that were not answered in this video, if you need help troubleshooting your experiment, or if you have a question about a CST antibody, please contact technical support at cellsignal.com/support. This will connect you directly to specialists with expertise in the assay. To learn about custom peptides and controls, products and kits for proteomic studies, and custom proteomics services offered by Cell Signaling Technology, visit the Proteomics Analytics Services page. Thanks for watching.